Welcome to Biology 110 at Agnes Scott College. This is the first semester of a two-semester introductory biology sequence. This semester covers foundational concepts in biology, including the building blocks of life, cell biology, Mendelian genetics, evolution, and ecology. Bio 110 is taught using a flipped classroom model. This means that the lecture usually given in the classroom is given as a voiced-over PowerPoint presentation that students can watch outside of class time and prior to the class related to this particular topic. In this manner, students come equipped with the necessary information for the day, and students are ready to engage the material through a variety of in-class exercises, case studies, simulations, and group work. This model builds the skills necessary for the expectations of nursing programs, clinical programs, graduate school, or medical school. It can take a bit of time to adjust to this mode of learning, but in the end, it will advance your science career. One of the benefits is that it puts you in control over the pace of the learning. Students arrive in college with varied backgrounds in their biology education, and a flipped classroom allows everyone in the class to engage the material at their own particular pace. As you go through these lectures, you are encouraged to pause the lecture, replay a section, look up things in your textbook or on the web. For the sake of studying, you should read the assigned textbook chapter first. Take notes on the chapter, then read through the lecture slides, and finally listen to the lecture. Use the assisted reading questions assigned to each lecture as a self-test of what you've retained from all your studying. There will be time in class for you to ask questions about any lecture material that did not make sense. Please utilize all the resources that are available so that you can be successful in Bio 110. There are student learning assistants assigned to this course, and their information is on your Canvas page assigned for this class. These learning assistants are a terrific resource. They hold workshops, drop-in hours, and can support you in your work in this course and in other courses. Your professors are also happy to meet during office hours. If those office hours don't fit your schedule, just reach out to the professor through email and set up an appointment. All right, let's get started. What you see on the slide is what's known as a concept map or an information roadmap or even a lecture flow chart. All Bio 110 lectures will have this concept map at the start of the lecture. Your faculty members have constructed these to help you compartmentalize or put in boxes the different topics and all their various parts to think about the connections between these parts. Today's lecture focuses on the key themes that will help you structure your thinking about biology. These unifying themes are evolution, organization, information, energy and matter, and interactions. We'll briefly introduce some of the core concepts that are associated with each of these themes, and we will go into much more depth as the semester progresses. We'll wrap up this introductory lecture by discussing the scientific process. During the lecture, we use both text and images to convey information. If it is on the slide, you are responsible for understanding and not just memorizing any information you see, for application questions both on quizzes and on tests. During the lecture, minimize your distraction by turning off any music, listening to the lecture in a quiet space, and turning off your phone. Take plenty of notes for lecture. Some students in the past have benefited from giving one page in a notebook for each slide and writing information that's on the slide down before they're even listening to the lecture. This is a highly effective way to ensure you're getting all the information before the lecture. Studies have shown that if you can change ink color in your notes for each section, it allows you to remember each section easier based on the color of ink on the page. Studies have also shown that if you handwrite your notes, you retain more information because you had to put the information in your own words. So let's turn off your phones. Let's get into a quiet space and get started. How do biologists study the astounding array of life? They do this by posing questions about the living world and seeking answers through scientific inquiry. Examples of these questions include: how does a single cell develop into an organism, which is shown in the figure on the bottom left? How does the human mind, shown in the figure on the middle, work and execute complicated behaviors and tasks? In the image on the bottom to the right, how do different forms of life in a forest interact? Biological life is often organized into various hierarchical levels. One way to think about this hierarchy is to consider the scale of study from large to small, or the other way around. The biosphere consists of all life on Earth and all places where life exists. The biosphere is shown as level number 1 in the figure on the bottom of the slide. The ecosystem, which is level number 2, consists of all living things in a particular area, along with the non-living components of the environment with which life interacts, such as the soil, water, atmospheric gases, and light. Notice that the picture associated with the ecosystem includes plants, animals, soil, snow, the atmosphere, etc. Level number 3 demonstrates a community. A community is an array of organisms inhabiting a particular ecosystem. Communities contain many different species that interact in a given location. The picture shows plants and animals, but don't forget that there are some that are many smaller, even microscopic members of the community that the picture may not show. Level number 4 in the picture shows a population. A population consists of all the individuals of one species living within the boundaries of a specified area. Here shown are lupine individuals. Individual living things are called organisms. So one of these lupine plants is an organism. The architecture of an organism is complex, but we can begin to break it down into hierarchies. A leaf is an example of an organ, which is shown in level number 6. An organ is a body part that is made up of multiple tissues and has a specific function in the body. Leaves, stems, and roots are all major organs of plants. When you observe a leaf under a microscope, you can see the tissue. The tissue is level number 7. Each tissue is a group of cells that works together, performing a very specialized function. Level number 8 is the cell. The cell is life's fundamental unit of structure and function. Remember that there are unicellular organisms, and so the cell is what is the fundamental unit that gives rise to behavior of certain organisms. The picture here in number 8 shows a magnified view of a cell in a leaf tissue. This cell in particular is roughly 40 micrometers wide. Within the cell, you can see smaller structures, which is shown in level number 9. The smaller structures shown in this particular picture are chloroplasts. These are organelles that are involved in photosynthesis. Photosynthesis is the process by which plants use sunlight to create energy and a food source to allow the plants to grow. The final part of this hierarchy is the molecular level. A molecule is a chemical structure consisting of 2 or more units. In this particular picture, they are represented as balls in the graphic. Overall, in Bio 110 and in other biology courses, we'll be examining various levels of this particular overall scheme at different scales. The study of life reveals several common themes, and these themes are really helpful to organize and make sense of all the information in biology. You will notice that these are not necessarily topics within biology, but again, they unify the ideas that span so many different discipline boundaries that exist within all of biology. This means that these themes are critical for understanding neuroscience and ecology. The themes are organization, information, energy and matter, interactions, and evolution. One way to challenge yourself is by connecting to the content in this course and then discovering which theme that particular content would fall under. And often it might fall under 2 or 3 different themes. As you think about biological hierarchy, you begin to appreciate the novel properties that emerge at each level. These emergent properties are a result of the arrangement and interactions of parts as the complexity increases. For example, although photosynthesis occurs in an intact chloroplast, it will not take place if chlorophyll, the key pigment, and other chloroplast molecules are simply placed in a test tube. An analogy for emergent properties is that a box of bicycle parts won't get you anywhere. A functional bicycle emerges only when all the necessary parts are connected in the correct way. We'll see a lot of examples of emergent properties over the course of this semester. In the image on the slide, we see how 10 levels of biological organization work together. Some courses study biology from the microscopic molecules, shown at level 10, to the macroscopic molecules at levels 1 through 6. Other courses move the opposite direction. For this course, after we cover some review chapters, we will move from the macroscopic levels, the ecosystems, communities, and populations, to the cellular and molecular levels of biology. Understanding the levels of biological organization will aid you in the lectures that follow, not only in this class, but also in your upper level sciences as well. So it is important that you are able to understand all the information that we talked about regarding this slide. Scientists often use a reductionist approach to study life. When we zoom in at an ever finer resolution, reducing complex systems to simpler components that are more manageable to study. Reductionism remains a key approach for biologists, but it is also complemented by systems biology, which is the exploration of a biological system by analyzing the interactions among its parts. The system might be a leaf cell, or a frog, or an ant colony, or a desert ecosystem. A systems approach means we can pose interesting integrative questions like, how do networks of genes in our cells function to generate a 24-hour cycle of wake and sleep? Or how does increasing carbon dioxide alter our biosphere? As we continue through this chapter, in the margins of your notes, come up with questions that would represent an example of a systems biology approach like the 2 we just discussed. Now we will cover evolution as a core theme in biology. Evolution makes sense of everything we know about living organisms. It explains the patterns of unity as well as the patterns of diversity in life. Similar traits among organisms are explained by descent from common ancestors, and differences among organisms are explained by the accumulation of heritable changes. The amazing diversity of life forms on Earth arose through an evolutionary process. These photographs show two different morphs of beach mice. They are members of the same species, but they look quite different depending on where they are found. The light-colored morph, shown in the photo on the left, is found on a beach habitat, and the darker colored morph is found on the right and found further inland where soil is darker and less sandy. The coat color of the individual mice tends to match their background. Why do you think this is the case? What is the function of color matching between the mouse coat and the soil in which they move? Did you come up with an answer? By matching the substrate that the mice live on, the mice might better be able to escape and being eaten by a predator that hunts by vision. An organism's adaptation to its environment, such as this mouse protective camouflage, is the result of evolution. Evolution is the process of change over time that has resulted in the amazing array of organisms found on Earth. Now that we've looked at evolution, let's talk about the next theme, organization. And it has two key topics that fall under this theme: structure and function, and the cell. At all levels of biological organization, you can find a correlation between structure and function. Because of such correlations, analyzing a biological structure can give us clues about what it does and how it works. For example, this hummingbird's anatomy allows its wings to rotate at the shoulders, so hummingbirds have the unique ability to fly backwards or over in place. While hovering, the birds can extend their long beaks into flowers and feed on nectar. This is a beautiful example of the match between form or structure and function as it is explained by the process of natural selection, which we'll cover later. So we're talking about organization, and we just talked about structure and function and how the two are correlated. The other part of organization that we're going to cover is the cell. The cell is the smallest unit of life that can perform all activities required for life. I'm going to say that again. The cell is the smallest unit of life that can perform all activities required for life. One way to remember this is the fact that there are single cellular organisms, little organisms that exist as a singular cell. We'll go more in depth in cells later in the course, but briefly, all cells share certain characteristics. All cells are enclosed by a membrane. There are two main forms of cells: prokaryotic and eukaryotic, which are shown on these diagrams. The eukaryotic cell is on the left, the prokaryotic cell is on the right. So we've talked about evolution and we've talked about organization. And under organization, we talked about the correlation between structure and function. And we also talked about the cell and how it is the smallest unit of life. Now we're going to talk about information. And under information, we're going to talk about DNA and the central dogma. Life's processes involve the expression as well as the transmission of genetic material. Chromosomes are a structure within the cell that contain most of the cell's genetic material in the form of DNA. Chromosomes are just DNA that's been tightly packed around some proteins and folded. A DNA molecule holds hundreds of thousands of genes, each a stretch of DNA along the chromosome. Genes are the units of inheritance that transmit information from parents to the offspring. They also transmit information as a cell grows and divides. The genetic information encoded by DNA directs development. In the images on the bottom of the screen, the DNA is shown in blue. Parts of the cytoskeleton are shown in red. These cells are in the process of division. As we again further explore the biological theme of information, we're going to talk about the central dogma. DNA provides the blueprints for making proteins, which are the major players in building and maintaining the cell. Genes control protein production indirectly, using RNA as an intermediary. You can see an overview of this process on the slide, and we'll go over this in more detail in a few weeks. DNA is transcribed to mRNA, messenger RNA, and transcription occurs in the nucleus because DNA can never leave the nucleus. The nucleus protects DNA. So DNA is transcribed to mRNA. And again, this takes place in the nucleus. mRNA, or messenger RNA, is translated into an amino acid chain, which becomes a protein. Translation of mRNA to an amino acid chain occurs in ribosomes, and this is outside the nucleus. So DNA is transcribed to mRNA. This occurs in the nucleus. The mRNA leaves the nucleus where it becomes translated into an amino acid chain, which becomes a protein. This entire process of DNA to mRNA to protein is called the central dogma of biology. So we've looked at evolution, we've discussed organization, and under organization, we've talked about the correlation between structure and function, and we've discussed how cells are the smallest unit of life. We've discussed information. We've talked about how DNA encodes for genes and resides in the nucleus in a packed form known as chromosomes. And we briefly looked over the central dogma, which is this idea that DNA is transcribed into mRNA in the nucleus. mRNA leaves the nucleus and goes to a ribosome for translation into an amino acid chain, which will become a protein. Now we're going to look at energy and matter. Moving, growing, reproducing, and various cellular activities of life all require energy. The main energy source is the sun, and the transformation of that energy from one form to another makes life possible. Plants and other photosynthetic organisms convert the energy of sunlight into chemical energy of sugars. This chemical energy from producers, plants or algae, is then passed on to the consumers that feed on the producers. Energy flows through ecosystems. It generally enters as light and exits as heat. Chemical elements are recycled within an ecosystem. Here's an example of these points. You can see the arrow indicating energy flow through the system. For chemical cycling, plants take chemicals up from the soil and the air. Those chemicals in plants are passed to the organisms that eat them, and then decomposers like fungi and bacteria break down leaf litter and dead organisms, and return the chemical to the soil. We've talked about evolution, organization, information, as well as energy and matter. Life does not occur in isolation. Instead, organisms interact with other organisms and their environment. So let's look at these interactions. The interactions between organisms and their physical environment can have a profound effect on the organisms and on the environment where the interactions occur. For example, a plant takes up water and mineral from the soil and carbon dioxide from the air. Or a tree releases oxygen to the air and the roots help form soil. The nature of these interactions can differ quite a bit. Some interactions can benefit both the interacting organisms and other interactions, both interacting organisms can be harmed. In further lectures, we'll discuss how we can classify these interactions between organisms. Here's an example of a mutually beneficial interaction between species. These fish feed on small organisms living on the sea turtle's skin and shell. The turtle benefits because the fish remove these potentially harmful parasites, and the fish gain nutrients from the meal and potentially some protection from predators by being so close to the sea turtle. Life on planet Earth and these large biological themes can be studied at different scales, from the molecular level all the way up to the biosphere level. We've talked about some basic examples of these biological themes: evolution, organization, information, energy and matter, and interaction. And while we know that these can be studied at different levels or scales, how do biologists study? We're going to discuss the scientific method and illustrate it with a case study using the beach mouse example. Science is a way of knowing. It's an approach to understanding the natural world. Inquiry or asking questions is at the heart of science. While you may have been taught that the scientific method is a formula or a set of rigid steps to follow, it is actually a more variable and fluid practice. Science includes elements of challenge, adventure, luck, careful planning, reasoning, quantification, creativity, patience, persistence, and a lot of teamwork. Although these factors mean that the scientific process isn't always superstructured, there is a process to inquiry that includes making observations, forming a logical hypothesis, and then testing that hypothesis. Collecting and analyzing observations can lead to important conclusions based on a type of logic called inductive reasoning. Inductive reasoning draws conclusions through the logical process of induction. Induction is where generalizations are drawn from a large number of observations. For example, the generalization that all organisms are made of cells is a conclusion formed by inductive reasoning based on 2 centuries of microscopic observations made by biologists. So induction is where generalizations are drawn from a large number of observations. There's also another type of logic called deductive reasoning. This is built into the use of a hypothesis in science. While induction involves reasoning from a set of observations to reach a general conclusion, deductive reasoning involves logic that flows in the opposite direction, from the general to the specific. From the general premise or the general inductive reasoning conclusion, we can extrapolate a specific result we could expect if the general premise was true. Deduction usually takes the form of predictions, the prediction of an outcome of an experiment. An experiment is conducted to test the hypothesis, and the results are compared to the prediction. Keep in mind that a hypothesis can never be proven. And I'm going to repeat that again. A hypothesis can never be proven. So as you go through science, you can say that your experiments support your hypothesis, but it can never be 100% proven. And that is because we can never test all the possible alternatives that could explain your data. So be careful with your language when you speak or write about scientific findings. We aren't proving or even disproving things. We are providing evidence for or against a particular hypothesis. As stated earlier, very few scientific studies actually adhere to the rigid sequence or steps that are typically used to describe the scientific method, which is often depicted by a linear set of steps. Here's a more realistic diagram of the scientific process, and we'll examine each component in a little more detail. In brief, you start with some exploration and discovery, some of that inductive reasoning that we were talking about. Then you use the exploration and discovery to form and test a hypothesis, again, some of the deductive reasoning. After you come up with some of your basic results, you'll go and use the community analysis and feedback. This is the scientific community. So your peers, your lab group, you're going to get some feedback to determine whether or not your overall method and your results are strong and sound. And then you can look at societal benefits and outcomes. Let's dive in a little bit deeper. Biology begins with careful observation, illustrated here by biologists using tools to aid in observation. Binoculars shown in the image on the bottom left, or microscopes shown in the image on the top right. These observations help biologists describe natural structures and processes. As they explore nature and the natural world, biologists rely heavily on scientific literature, which includes the published contributions of fellow scientists. By reading about what is already known, scientists can build on that foundation of existing knowledge and focus their efforts on tackling the unknown. Exploration and discovery shapes the central goal of forming and testing a hypothesis. So the exploration and discovery represent the inductive reasoning, looking at all the data, considering all the information out there, forming a hypothesis, again, representing the deductive reasoning. Most scientists would argue that while you do a lot of this exploration and discovering prior to hypothesis formation, this process in reality is almost always ongoing. In science, a hypothesis is a rational accounting for a set of observations guided by inductive reasoning. In plainer English, a hypothesis is based on observations and assumptions and leads to a testable prediction. You can think of a hypothesis as an explanation, but an explanation on trial. The hypothesis leads to predictions that can be tested with either additional observations or an experiment. An experiment is simply a scientific test, and is often carried out under controlled conditions. Although the scientific process is quite flexible, forming and testing a hypothesis is central to this process. This part of the process involves testing ideas, forming hypotheses, predicting results, doing experiments, making observations, gathering data, and analyzing the results. Interpreting the results can indicate that the data support or contradict a given hypothesis. Both can inspire a revised or a new hypothesis. Let's use a very simple example to illustrate how this overall scientific method works. You make an observation that when you turn on your desk lamp, the bulb doesn't light up. You make a few other observations, some general inductive reasoning. Your lamp is plugged in and the power is currently functioning in the building. These are several observations and again are part of that inductive reasoning. Our obvious question in this simple example is, why doesn't your lamp work? There are two reasonable hypotheses here. One, the bulb is burnt out, and two, the lamp is broken. Both of these are testable hypotheses. You can form predictions here as well. If the bulb hypothesis leads us to predict that changing the bulb would then cause the lamp to turn on. The lamp is broken hypothesis could lead us to predict that even changing the bulb would mean that the lamp wouldn't turn on. These hypotheses can then be tested simply by changing out the light bulb, and this changing out the light bulb is our example of an experiment, the scientific test. And we could carry this out under controlled conditions by checking our other lamps and maybe even changing out their bulbs as well. The factor that is manipulated by the researcher is the independent variable. And the effect on the independent variable on any system are considered experimental variables. The effect of the manipulated factor is called the dependent variable. Researchers usually control unwanted variables not by eliminating them, but by canceling them out using control groups. The scientific process is highly collaborative, and it's one that's influenced by interactions with other scientists as well as with society. Examples here include technology development, science informing policy, problem solving, and building a shared knowledge base. Scientists hold their work to a high standard, and they rely on feedback from peers. In order to publish results of a scientific study, the manuscript describing those findings is reviewed by several experts in the field. Additionally, scientists often seek to replicate each other's work to provide additional support for the hypothesis in question. This builds scientific consensus. Science is inherently a social process. While scientists are often depicted as people working in isolation in the lab, most scientists work in teams, which often include graduate and undergraduate student researchers. Science benefits from the work of those who have performed experiments and made observations before us. And scientists in the same research often check each other's claims by repeating experiments. The research community is part of society at large, and their relationship of science to society becomes clearer when we add technology to the picture. The goal of technology is to apply scientific knowledge for some specific purpose. Because scientists put new technology to work in their research, science and technology are interdependent. So today's lecture covered 5 big themes within biology, provided a few examples of concepts and topics corresponding to each of those 5 themes, described the hierarchy of biological organization, and introduced the scientific process. If you have any questions about this lecture or the additional information in your textbook chapter that supports this lecture, please reach out to your professor through email or office hours, or feel free to bring your questions to class. Additionally, consider visiting the learning assistants to clear up anything that did not make sense in this lecture. As lecture is over, go back and double check your notes, integrate any information from the book, or from outside sources that help you understand what you've heard today.